Our next speaker is Dr. Ken Hayworth. Ken is a senior scientist at the Howard Hughes Medical Institute Janelia Research Campus. He's also a longtime friend of mine. Ken has a long history of research directly aimed at making whole brain emulation a possibility. For example, Ken invented and developed the automatic tape collecting lathe ultramicrotome to very finely and carefully cut and store slices of brain tissue, creating what amounts to a library of brain slices for imaging and study. He's also the president and co-founder of the Brain Preservation Foundation. Ken is directly responsible for the Brain Preservation Prizes, two of which have by now been awarded, most recently the Large Mammal Prize. I'm sure he can tell us all about that, and at this point, Ken, I'll let you share your screen and take over from here.
Okay, can everyone hear me?
Yes.
Great. Okay, I am sharing my screen. I assume that went well. Randall, thank you very much for inviting me to give a talk at this conference. I want to make clear, crystal clear, up front that I am speaking as the president of the Brain Preservation Foundation, and I am in no way giving views that should be associated with any of the scientific institutions that I have been employed at. In general, I am speaking on my own behalf with my own, sharing my own ideas. So with that preamble, what I want to talk about in this talk, and it's going to be a very brief talk. I really feel like I'm unclear at what level people are coming at these questions from. So I'm going to make this a very short talk so that we can hopefully open it up for discussion and debate and get to what people really are interested in. And I'm gonna talk a little bit about what Keith just talked about. He was absolutely brilliant and clear. I'm going to be less of both. But I really think before I dive into anything, I've had a lot of debates and discussions with neuroscientists and with others in the scientific community about brain preservation and mind uploading, and unfortunately, there is less of a debate about what we should be debating, which are the technical issues, and more of a blockage due to this notion of what consciousness is. For example, I think if you were to summarize the debate within the neuroscience community, it's really a debate between those people who think that the neuroscience community is going to be successful ever in its quest to get a complete computational understanding of the brain, and those, for whatever reason, that think that a complete computational understanding of the brain is simply impossible. I know it's hard to believe that some neuroscientists feel that way, but from my discussions, I really have gotten the impression that quite a few do. The other side of that, or the other part of that debate, is there are people that there are neuroscientists that think that yes, we may be successful in the infinitely far future, but the progress that we've made so far in terms of understanding the brain is so minuscule that we are not even allowed to debate this issue. I have run into that quite a bit. It is quite frustrating because, if I'm looking at a set of neuroscience papers that say this is how memory is encoded, this is our models of how the brain works, et cetera, and somebody says, well, I agree with all of those, but we have no idea what consciousness is, therefore, we can't even start a discussion about what it would require to preserve a brain. To me, that is really showing that they don't have a scientific objection. They have a philosophical objection that probably fits somewhere within the list of philosophical objections that Keith just described. So in any case, with that preamble, let me switch to the first slide here. What I want to address is what are the structural and molecular features of the brain that must be preserved to make possible, at least in principle, the revival of the person via future mind uploading. And the ground rules, we are assuming physicalism. The brain is solely a product of computations in the physical brain. I would argue strenuously that all of neuroscience is based upon this assumption, and that there's 100 years of neuroscience and neurological experiments, from Phineas Gage to split brain to all sorts of experiments that support this fundamental conclusion that the brain is computation, that the mind is computation that the brain does. We are assuming that any brain preservation technique like the aldehyde-stabilized cryopreservation that I'll be talking about, that it allows the brain to be stored essentially indefinitely. So we can be free to assume as advanced a future mind uploading technology as we want, within reason. So I think we should be free to assume that in however many hundreds of years that we will have figured out how to produce nanometer scale electron micrograph images of entire human brains, that will be able to annotate those structural images with molecular information about the identities and locations of ion channels and receptor proteins, et cetera, et cetera. And so this really boils down to a question of whether sufficient information is being preserved, and that's the real question. We accept, of course, that we're talking about brain preservation, but we really maybe should be talking about whole body preservation. There's no reason why an ASC based technique or some other one, if somebody invents it, couldn't preserve the spinal cord, the retinas, the body, etc. But at the same time, we realize that even current society accepts that somebody with a spinal cord transaction is still the same individual. And we accept, of course, that current neuroscience knowledge is incomplete, fantastically incomplete. So any answer to this question must come in two parts. Do we know enough to even speculate about what would be required? And given our current understanding, what would likely be necessary to preserve? So, touching on the philosophical issue again, I think one thing that I disagree with, if you will, in what Keith said, and I doubt it's a real disagreement. It's just probably terminology. Is I think when I look at a question like, is it possible to revive a person? Is it possible to mind upload a person? The immediate thing that a neuroscientist should say to that question is that there is no person. There can be no hard scientific answer regarding the success of the revival of a person, because we know that the traditional folk psychological concept of person is fundamentally incompatible with our computational view of the mind. For example, we could ask a similar question regarding any software program. For example, what data and algorithms must be implemented to revive the classic arcade game Asteroids on a new hardware platform. I hope it's obvious that in this context that good enough is in the eye of the beholder. For example, some will insist that any Asteroids game must use vector graphics to really be called the real Asteroids, while others would be very happy with raster graphics like the Atari used. There's no true answer to this question. Well, if you really believe that the mind is computational, and again, that's the foundational core of neuroscience, if you really believe that the mind is computational, then you must also accept that there is no further fact, as Derek Parfit would say, as to whether an imperfect copy of you is really you. So what about continuity of consciousness? To elaborate a little bit more on what Keith said, we are animals with mechanistic brains that have evolved to intelligently control our behavior in a complex environment. Most of the brain processes operate unconsciously, but humans have evolved a self-modeling function that allows our brain to interpret its perceptions, goals, actions, etc., to form an internal narrative about a self that has a point of view, that is experiencing the world and has internal feelings. It has wants, it has goals, it has beliefs, it makes decisions, and it takes actions. This self-model narrative is continually being written into our long-term memory in the form of changing synaptic connections, and is used by the brain to help better plan for future behaviors. The key thing to understand is that this self-model, and this is the dominant theory in cognitive science. If you ask a cognitive scientist what is the default theory of how consciousness works, they're gonna say this. They're gonna perhaps talk about global workspace models and left hemisphere interpreters like Mike Gazzaniga. There's a lot of details that go into this. There's a lot of neurological studies that have gone into building up this model. But in any case, this self model literally describes what it is like to be us. That's the type of information that is written into this self model. And it feeds back on itself to create our consciousness and ongoing sense of self. So at a fundamental level, all there is to consciousness and self is this. And I'd be happy to debate this later. But when we are in a deep sleep or under general anesthesia or falling into a frozen lake, as Keith was mentioning, all of these processes are obviously turned off. Our consciousness ceases to exist. And when we wake up, we feel that we are the same person that went to bed last night only because our brain's long-term memories that allow us to recall previous experiences and hook them into our ongoing self-narrative. So there is no true continuity of consciousness in the folk psychological sense. There is only data and information. And I feel like this is absolutely obvious, and it should be absolutely obvious to anybody that is in the neurosciences and cognitive sciences. And yet this is the objection that keeps coming up over and over and over again to the idea of mind uploading. So in any case, I'm okay with this model of who we are, and I think everybody should be okay because it doesn't take anything away from our humanity. It just helps us understand the neurobiological underpinnings of what we feel is important. So having said that, many people that do embrace this scientific model of that the mind is simply a product of neuronal computations tend to seem to agree on what would be the crucial features that that make us us. Long-term declarative and episodic memories. These are what make us believe that we're the same person today as went to bed last night. These are also the types of things that differentiate me from other individuals in the society. And other forms of learned and innate knowledge, skills, personality, et cetera, et cetera. These are the things that make us unique individuals. And so there is strong evidence that all of these are stored in the pattern and strengths of synaptic connections. If we were to look at a computational level, and I think the strongest objection that I have to Randall's talk, for example, is that I never hear a computational level. What is the computational level of description? That abstraction from the neurobiology that says, it's not really the neurons and their spikings that are important, it's X. What is that X? Well, maybe I should take a second right here and say what I think that X is. If you look at neuroscience experiments, so, for example, if you look at experiments where they record from either human or monkey brains and look at the representations of faces, there's a great paper recently where they went in and did multi-unit recording on a monkey face area and determined the code for representing faces in that particular monkey. What they needed to do is they needed to look at the patterns of firing of neurons in that particular area. That's what represents a face in that monkey brain. If we look at at experiments in mice that are looking at the hippocampal place cells and looking at the patterns of neuronal firing in the hippocampus that represent not only where the mouse is currently, but will represent sequences to say where it might be going, almost like it's imagining different paths that it can travel in. When you look at those experiments and how they decode things, they're decoding patterns of neuronal activity. And they're decoding not even just patterns of neural activity, they're decoding what are called neural ensembles or cell assemblies from Hebb's original terms. These neuronal ensembles are groups of neurons that fire together that mean something. And we know that they are connected. These groups of cells are connected together by excitatory connections in the cortex. There's even connectomic research that has shown this. These groups of cells are the things that signal to other groups of cells that they should turn on or off. These are the states of different brain regions that are forming a computational level of the brain. And so if we really look back and say, what is, if what we really are is a computational system that does a whole bunch of computations unconsciously, like recognizing faces and deciding to approach or withdrawal, and if we look at that, and then we say there is a self model that is representing on top of that what those decisions were, and those are being encoded in memory, then you can see that the representations that matter are patterns of neurons activating other patterns of firings in neurons. And what encodes memory are synaptic connections between those patterns of neurons. And so let's see where I'm going with this, because I think I'm probably rambling. So you might argue that, and I have argued in the past, that an electron microscopic map of a preserved brain would be sufficient to decode these types of neural to neural interactions, is specifically episodic and declarative memories and procedural memories, et cetera, are thought to be encoded by very particular types of synapses in particular areas of the brain, and the models seem to suggest that we should be able to decode those, to be able to see the states, what states an area can fall within, can fall into, how those states interact with each other. It seems like it should be possible to decode all of that from an electron micrograph image. But let's assume for today that that isn't possible, because there is reasonable scientific debate about that. For example, you might know that a neuron has a certain level of connections with other neurons, but if you don't know its electrophysiological properties, what ion channels it has, you may not know its threshold of firing. You may not know the strength of a synapse may be correlated, as we've seen in many, many studies, it may be correlated with the size of a synapse, but maybe it's not perfectly correlated. Maybe it's not correlated enough. We know that fundamentally the strength of a synapse is determined by the number of, for example, an excitatory synapse in the cortex is determined by the number of AMPA receptors. And so if one had molecular level information about ion channels and receptor proteins that were in addition to all of the structural connectivity of the brain, then it seems to me, almost on the face of it, that this should likely be able to preserve the memories, or be able to, future neuroscientists should be able to decode these types of memories based on such a map of connections and ion channel and receptor protein information. So the answer to this question then, I suggest, is that the morphology of neurons and glia, the patterns of synaptic connectivity, the ultrastructural details of synapses, the ion channel and receptor identity locations, those are the things that go into large-scale neuronal modeling today. These are the things that should be sufficient because they form the causal pieces of neuronal processing over time frames, seconds to minutes, et cetera, that we typically associate with our thoughts. The fact that we can retrieve a thought about our loved ones in just a few seconds pretty much means that the ability to retrieve that thought did not require new protein synthesis, and it did not require us to analyze the methylation pattern of our DNA. These types of time frames that our thoughts occur on really have to be based on electrophysiological properties of neurons and synapses. So with that, it appears to me that aldehyde-stabilized cryopreservation, this new technique, can preserve all of these structural and molecular features, at least in ideal laboratory animal demonstrations. So I'm about to go on into what ASC, how ASC works, but I want to pause to see if there's any particular questions at this point, and to see if anybody's been listening to me. Randall, it'd be nice if you.
So if you can hear me, I certainly have several questions. I'm not sure how you are with time, because let's see, it's now.
So I want this to be very open. So I am fine with time.
Okay. You're not worried about the rest you still have to tell us? about ASC or whatever.
Yeah, I would rather you drive the conversation. I can give a explanation. How about I do this? I can give an explanation of what ASC is for anybody that might have dropped in. I can make that sure that's like a 5-minute explanation, and then I would prefer to just open this up to questions.
Okay, let's do that. Okay, I'll just mute myself while you give that explanation, and then we'll get right back to questions.
Okay, so so as Randall mentioned, about 7 years ago, I founded an organization called the Brain Preservation Foundation because I wanted the scientific community to reevaluate cryonics. I said, cryonics is a fantastic idea. It was invented 40 years ago. People say that it's still impossible, but I haven't seen any scientists really reevaluate what the current state of the art is. So I found a donor, put forward a challenge prize not only to the cryonics community but to the scientific community to say, look, the standard objection to cryonics has been for decades that the structures that encode memory in the brain are being utterly destroyed by cryonics. I knew that wasn't necessarily true in ideal cases, but I wanted to make sure that that there really was a high standard that was being met. So I said, this challenge prize, this brain preservation prize, demands open scientific proof along with publication of a technique on laboratory animals in ideal conditions that show that you can preserve the synaptic connectivity of an entire large mammalian brain, and that you can preserve that for at least 100 years. Demonstrate a method that you can see is essentially able to preserve for over 100 years. And so the long story short, the cryonics community was not able to meet those stringent requirements. That doesn't mean that they destroy the brain in all cases. But it means that for some reason or another, they are unable to produce a publication that can show that the connectome has been preserved. So it's still an open challenge to them, but I guess unfortunately for them, in the meantime, a different technique that does not involve or that is not standard cryonics was able to win the prize. And this different technique was developed at the Cryobiology Research Institute called 21st Century Medicine. And let me go into that. So the ASC, Aldehyde-Stabilized Cryopreservation, was invented by Robert McIntyre and Greg Fahey and published first in this 2015 article. The ASC technique is actually extremely straightforward. It's so straightforward that I think it might be the best for a while. I don't see anything getting better than this anytime soon from a structural and molecular preservation of whole brain, from an information preservation point of view. What they do is they've essentially taken what is standard practice for the best electron micrographic research of the brain, which is to perfuse the brain's vascular system with the deadly chemical fixative glutaraldehyde. So clearly that kills everything, which is good because you want to stop the decay process as fast as possible. What glutaraldehyde does is it covalently links proteins together at particular amino acids, and that locks them into position and prevents them from attacking each other and causing decay. They have combined that glutaraldehyde perfusion with the next step in the procedure is they introduce a cryoprotective agent into the fixative, and ramp it up to about 65% over the course of about 4 hours. What that means is that at room temperature, they are able to continually perfuse cryoprotective agent in a not a viscous form because it's at room temperature, so that they can reach a uniform high concentration of cryoprotective agent throughout all the tissues of the brain. Once that uniform high concentration of cryoprotectant is introduced, then the entire brain can be just put into a freezer. And when it goes below minus 120 degrees Celsius, it solidifies. It does not form ice crystals because of the high cryoprotectant agent concentration, but it solidifies and prevents all further diffusion and chemical reaction. And essentially, time has stopped for that brain. So essentially, the ASC procedure has combined the best in chemical fixation with the best in cryopreservation. So I just want to remind everybody that all of the recent connectomics papers, the large-scale reconstructions of any type of neural circuitry, really starts with glutaraldehyde perfusion fixation. So you can look into the neuroscience literature and see that glutaraldehyde perfusion fixation of brains is being used everywhere where you want the synaptic connectivity of the brain and the ultrastructural details of synapses to be preserved. What is less commonly understood is that glutaraldehyde fixation does a very good job of preserving the identities, if not the biological functionality, but the identities and locations of proteins. And so, if the key point is that that glutaraldehyde is the best method to preserve both the structural connectivity of the brain and the molecular level of information in the brain if you want to do this over an entire brain scale. There are other methods if you're trying to get only a cubic 100 microns or something. You can use high pressure freezing or something like that. But if you're looking for a method that does something like a whole brain, this glutaraldehyde perfusion is the best. And again, all that ASC is doing is providing a way to store a glutaraldehyde perfused brain for an indefinite period of time at low temperatures. So that's a ASC brain going into a freezer, and this is the same brain. It's a rabbit brain in this case that's taken out of the freezer the next day, and you can see it is a solid block. So it could stay in that shape forever, essentially. That brain was taken out and sliced up and prepared for electron microscopy by staining slices with heavy metals and plastic embedding those slices. And it was evaluated to look for gross dissection, gross problems with the brain, which were not seen. And it was evaluated at the ultrastructural level as seen here. And this is a video. Hopefully it's coming through without too much artifacts. This is a beautiful FibSim video of a piece of that rabbit's brain's cortex, showing all of the synaptic and neural details have been preserved. There's no ice crystal damage that is seen. It looks very much like a control, just glutaraldehyde fixed brain. This same technique was applied to a whole pig brain. And you can see in these images that after rewarming, these are all after rewarming of this pig brain that had been stored at minus 135 degrees Celsius in a solid state. After rewarming, it was dissected to look for gross damage to the brain. We don't see any ice crystal damage anywhere. And it was also put through a rigorous testing in multiple places to see what the ultrastructure of synapses looked like, and it looks just as good as any brain that is traced for neural connectivity in connectomics studies. So a quick summary, what does ASC preserve? Well, ASC is simply glutaraldehyde perfusion fixation of the brain combined with indefinitely long-term cryostorage. So it should preserve everything that glutaraldehyde does, but for centuries if necessary. ASC has been proven to preserve the structural connectome. ASC has been proven to preserve the ultrastructural details of synapses. The volume of synapses, the synaptic area, the number of synaptic vesicles, etc. And these are all thought to be correlated with functional strength of the synapse. ASC should preserve the locations and primary structures of key neuronal proteins, for example, ion channels and receptor proteins. Why do we think that's the case? Because glutaraldehyde preserves these structures, and the cryoprotectant is mostly inert from a chemical perspective. And so whatever the thousands of studies that involve what glutaraldehyde preserves should, for the most part, be relevant to what ASC preserves. So what doesn't ASC preserve? Well, proteins are cross-linked, that rendering them biologically inactive. Again, the brain is dead. Obviously, you perfuse the brain with glutaraldehyde, it's dead. Fragile molecular states would be disrupted. Small molecules that are not cross-linked will diffuse away. And extracellular space is typically lost during fixation. And since lipids are not fixed by glutaraldehyde, in some samples I've seen membranes can be somewhat distorted. So the other advantages of ASC, only the initial perfusion of the brain is time critical to avoid ischemic damage that could cause vascular blockage. But all other subsequent steps, this is very unlike cryonics, all other subsequent steps can be extended essentially indefinitely to ensure that a uniform high concentration of cryoprotectant is present in all brain areas. So once the brain is glutaraldehyde fixed, you really can take your time to make sure that the cryoprotectant is saturating all of the brain tissues. ASC storage is resilient to short-term rewarming. When rewarmed, ASC preserved brains are in an optimal state to undergo the types of tissue preparation steps that are likely necessary for brain scanning and mind uploading, at least from our perspective today. They can be reperfused with lipid fixatives, heavy metal stains, and there are quite a few papers that show that at least a glutaraldehyde fixed brain tissue can be immunostained to look for to label particular classes of proteins. And so the key point to remember is that ASC simply stops time at the glutaraldehyde fixation stage. And I think I'll open it up for questions at that point.
Thank you very much. We've been not clapping for people so far, but I think we really should. So I'm going to clap right now. So as far as questions are concerned,
that was for you.
Yeah. Sorry. I'm going to be asking you questions, and they're going to come from 3 different sources. One of them is just questions that I have. The other one is questions from the room here. And then finally, there are questions from the forum where people remotely are submitting questions. But I should note that I haven't seen any remote questions come in a little while, even though the live stream still says that there are definitely a couple of dozen viewers at this moment. So I'm not sure if it's just because no one has any questions in terms of the remote viewers, or maybe they waited until you were done and now they're coming up with their questions. So I'll mostly be doing mine and the ones from the room. And I'll open it to the room first. Does anyone here have a question for Ken? Yes. Would you like to also state who you are? Well,
Hey, sure. This is Lou Sheffer. Hi, Ken.
Can he hear you? Yeah.
Hi, Lou.
I work with Ken. How are you? So the question I would have is, even assuming the glutaraldehyde works, can you read out all the things you need at the same time? Because one of the traditional problems is that when you stain, say, with osmium to show the membranes, you clogger the proteins that you might need to get the memory. And so the question is, can you read out both the proteins you might need for memory and the membranes you need for connectivity at the same time? So maybe the glutaraldehyde worked, but now you have a readout problem from the glutaraldehyde to the final representation.
Yeah, I think that is a question that we have today, because we have to live with the current technology that we're given, which is electron microscopy and a limited amount of data. I'm getting a lot of feedback.
Maybe I should mute while Ken is answering. Okay.
So whereas I agree that it is a particularly relevant question for all of the connectomic studies that are done today that might want to prove that that it would be possible to emulate a part of a small brain. If we're thinking about brain preservation from a future point of view, I have to think that if those features are needed, that future technology will get around the types of limitations that I think you're describing, Lou. So let me try to crystallize what I suspect your question is. So if we have a glutaraldehyde perfused brain, we can either decide to put it through osmification steps, plastic embed it, slice it up, and look at it under the electron microscope, and get really detailed images of the ultrastructure. But by putting it through that process, we have basically destroyed our ability to do good immunostaining on that. So we can't figure out where potassium channels are and what the density of potassium channels are on this particular neuron, and that might be really crucial to understanding what its threshold for firing is. Whereas we could take that same brain and we could put it through a different process. I'm putting my hand over here. We could put it through a different process and immunostain it, and we'd get really good images of where those potassium channels are. But now we would have, by those steps, removed a lot of the lipid information, and we couldn't go through the electron microscopy as well as we were before. And if you multiply this out to say, look, maybe we need not just the potassium channels, but we need these NMDA receptors, and we need these other classes of metabotropic receptors, and all of these things that are really preserved in a glutaraldehyde perfused brain, and by assumption in a ASC preserved brain, we essentially have this problem of yes, it's all there, but we can only read one at a time. And what I'm saying is, yes, that could be a real question. But if it is a real question, then that might mean that nobody gets uploaded for 200 years instead of 100 years. Or nobody gets uploaded for 500 years instead of 100 years. Does that address your question?
Because we needed this in.
Now you can hear you. Yeah.
Okay, yes, that's the question I had. And I think you answered that question. Now, of course, there's also a question of whether there's some subtle part that you need that the glutaraldehyde destroyed. But I think in general, I agree with you that probably it doesn't. But we don't know. Maybe there's some, like you said, maybe there's some real tricky methylation step that's involved in the long-term synapse strength. For example, in the fly, at least, we know the synapses that we see that contain memories in the mushroom body don't look any different to us. So we don't, by EM, so we don't know how the strength is stored. maybe the glutaraldehyde doesn't kill it, but
Well,
we don't know that for sure.
Exactly, and this is why I just read incessantly the neuroscience literature of how memory is stored in the hippocampus, how memory is stored in the striatum, how memory is stored in the cortex, in the amygdala, and in all of those different regions, there are good studies that have looked at the excitatory that have said, look, the crucial synapses that are thought to store information are excitatory synapses, glutamatergic synapses onto dendritic spines of principal cells, and those synapses, those spiny synapses, unlike the ones that you mentioned in fly, those spiny synapses, there is a ton of papers that show that their size, their literal size, the volume increases when their function strengthens. And the information that that has given that has come from a lot of different points, and so whereas I think this is the most exciting aspect of the discussion, that what really is necessary to get at the information and to emulate it. What is the computational level? What I would say is, okay, look, we should go into, if we go into the hippocampus, what we're going to see is a bunch of neurons that are cross-connected with excitatory connections. We're going to see those excitatory connections. Some of them have large synapses, some of them have small synapses, some of them have no synapses between them. And if you were to map out the way that that connectomic studies are doing, you would see collections of cells that are obviously forming a unit that means something to the animal. Maybe it's a place in the environment, and other collections and these would just pop out directly from the electron micrographs. I believe that is what we're going to see. I look at stuff like the Wei-Chun Lee and Clay Reed's paper in the visual cortex that came out in 2017. They saw essentially neuronal ensembles, if I'm interpreting the paper correctly, that these cells fire together and they have stronger excitatory connections between them. So they've seen some very big hints of this in connectomic studies already. I think that as connectomics grows, we'll see that you really don't need all of that ion channel information to get to the real computational level that is forming the mind. But if that's not the case, ASC is covering, I think, and I want to make the strongest statement possible. I want to make the strongest statement possible, so somebody will debate me here. ASC is covering all of the neuroscience models that exist today. That's got to be too strong of a statement, but I'm going to throw it out so that somebody jumps on it. So give me a neuroscience model that ASC doesn't cover, please. Great. Person in the back.
I wasn't going to give a model. What I was going to see is this is just framing in my mind, but it seems to me there's a little bit of circular thinking involved. Everything that we know about memory, or most of what we know about memory, and memory at the small scale, we know from electron microscopy. Therefore, the we assess ASC by electron microscopy, but isn't that only?
So I would disagree with the first part of that statement. I think most of I'm big on electron microscopy, but in terms of where it stands in the neuroscience toolbox, I would put it at a pretty low level. I think the Tonegawa experiments, where they tag memory cells and then reactivate them optogenetically, and can use electrophysiology to determine their connectivity, the connectivity among ensemble, not ensemble, engram cells, what they call engram cells. They can determine all of that electrophysiologically. There's another paper it's a fantastic paper where they and again, no electron microscopy. They developed a method. It's in the Kasai lab. They developed a technique to whereby only those synaptic connections in the motor cortex of a mouse would become labeled with a photoactivatable version of the RAC1 protein. And what that meant is that it could this genetic technique could literally light up only those synapses that were involved in a particular motor memory, in this case, a mouse that was running along a beam, that had learned to run along a beam. And then they could, at a later date, after it had learned a different task, could go back and shine light on those synapses and get them to shrink. And the mouse forgot its learning in that task, that motor task, but not the intervening task that had learned. So this has nothing to do with electron microscopy, and yet these are experiments that are really truly showing that it's the connectivity and the sizes of synapses that are encoding that information.
I think the problem here was that I temporarily forgot the word electrophysiology.
Okay.
So yeah, no, I'm right behind you. I understand completely.
Great.
Yeah. You could have answered it with one word.
Okay.
Okay, Ken. There are now also some questions coming in online, so we might switch to those in a second. But I'm gonna try to at least give you something, because you're asking for someone to say, well, what in our models of memory is not contained or not preserved by ASC?
Yes.
And to be honest, so I have no problem at all with your assumptions about what are probably the abstraction levels at which we should be looking for the computational model, et cetera. So you mentioned at one point that you had an objection with my talk at that, but maybe it was just that I wasn't setting a specific abstraction level. I was just throwing out all these possibilities, right?
That's right. That's exactly what I was,
right.
That you weren't setting a level.
Yeah, because I tend to agree with the ones that you mentioned. But see, when you say what is not being preserved by ASC, and I think that's what my talk later on is going to get into a bit, is it's not so much the evidence, the information that you can find in a brain at any point in time, but it is about other things that you do when you're creating a model, when you're reverse engineering what is there, the other information that you need to have. And of course, there are lots of kinds of information that you can gather by knowing about other brains, by having generic information about the brains of human beings. But what I'll ask in more detail later is do you really have everything you need for that? And I'm coming at it as a modeler, as someone who knows that every time you build a model, that first model is never correct. And it's not just not correct because you weren't careful enough or because your technology for making measurements wasn't good enough. It can be other more fundamental problems as well, such as, for example, the methods you're using, the algorithms you're using to estimate parameters or that you're using to select your models may not be the sort of algorithms that only produce one possible solution. And so at that point, you have to have some way to verify which solution is the correct one. So this is what I would say is the risk of assuming that everything is preserved in that piece of brain, no matter whether it's ASC or any other technique. But as I said, I'll talk about that for like an hour or so, and we'll have a discussion about it later. So you don't have to explain all that right now.
Well, let me add. I certainly agree. Although I think we probably would weight the possibilities differently. let me throw out a little bit more controversial of a question back, if I can. Can people hear me?
Sorry, Ken. It looks like the something happened over on Javon's side.
So often it gives an error message and then it goes back live.
Now we still can't hear Ken though, or at least I can't at the moment.
Testing.
He might have muted himself. I'm not sure.
Did you mute yourself, Ken?
No, I can see my the green bars going up and down on Hangout. Testing. Can anybody hear me on the Hangout as well? Alicia does. Okay. Wow, I should say something to the live stream that Randall can't hear.
Okay, you can probably still hear me.
Yes,
Javon
I can.
thinks that the problem may be on your end. Can you
end.
say something right now?
Testing.
Okay, I'm hearing him coming out of somewhere.
I know, it's not on his end, it's on ours.
It's on our end. Okay.
Comes out the earphones, but not the speakers.
No, the earphones are not plugged in anymore. It was coming out of some small speaker here in the machine right now, instead of the big one that we've been using before.
Testing.
That's so odd.
But we can fix that because I brought some speakers just in case.
It wasn't coming out of the television.
Try stop plugging the USB and plugging it back in.
Yeah, I did that.
So it's like Nintendo, yeah.
Right.
Instead of the cartridge.
Would it help if I just plug in a speaker that has a regular speaker sort of thing?
This is that supposed to be a regular speaker, right?
So it's coming out of that thing?
It was coming out of that thing earlier.
Can you say something, Ken?
Testing.
I'm still hearing it from here.
Yeah, it's coming out of the speakers on this laptop.
But what if?
Oh, it's just LG. Let's just change this one real quick.
Well, it must be that thing because
Yeah.
it actually, this thing was fully connected and working, and now it's just on.
Okay,
his
that's
camera.
working now. We should be able to hear him now.
Can we?
Testing.
Okay.
Okay.
Great.
All right.
For some reason.
I'm gonna mute myself while you speak.
Great. So I was going to just add one controversy. So essentially, what you're saying, and I hear you. It's a very detailed argument in terms of maybe all the information is being preserved, or maybe some fraction of the information is being preserved. But are you actually going to be able to use that information to successfully build a model because of all the details of model building that we know has a lot of parameters in it? And I have so much to say on that in terms of how I think the brain works, but actually I'm not going to say that. I want to just leave with one controversial statement. What should the criteria be for a brain preservation technique to be allowed to be developed and deployed in hospitals? Because if I'm and this will happen, if I'm dying and I have only a few months to live and there is no way out, and I have a chance to go to the local hospital and get a high quality regulated brain preservation procedure using a technique like ASC. And some neuroscientist says to me, you can't, because I have some really kind of fringe theory that, if it's true, might not work. Means that you might not be able to come back. That's going to ring very hollow with me, right? Because if all of our current theories are suggesting that, yeah, it probably will work, then it seems to me like that's already reached the threshold of developing it into a medical procedure. And so what I'm looking for is a real open, vigorous debate within the neuroscience and medical ethics community to start to say, what more needs to be done? What more needs to be said for this to be developed? I can think of a whole bunch of things that do need to, but I think the idea that there might be some memory theory that we haven't discovered yet, and that might not be compatible with preservation by ASC, that is not a sufficient argument to say to this terminally ill patient that they should essentially just suck it up and die, rather than having the chance of being revived in the future.
Thanks. Yeah. So we probably have time for just one more question, and I think I'm going to try to formulate one that sort of takes what you just said, but what is the procedure that would be sufficient, and what about these arguments that are more from some sort of belief or theory about that there may be something we don't understand, and also the question of what can you provide to someone in a hospital. I think you're right. It's strange to think that you would hold something back just because there's someone out there who believes that there may be something we haven't figured out yet, and that therefore we shouldn't do anything at all. It's kind of like saying, if we haven't figured out the actual way to cure some disease, but we have figured out something that will probably alleviate symptoms, then why not? Yeah, you're not allowed to use this drug because we haven't figured out the complete cure yet, or we're not 100% certain of how it will work and whether the patients will show some other side effect in 10 years. But there is, of course, this question about what sort of claims you can make. And this ties into a question that came in online, because there was a question, again, by Leslie Seymour, any commercialization availability status info compared to the existing cryopreservation practices. In other words, he wants to know, where is this going? Is there going to be a commercially available ASC anytime soon? And I'll sort of attach to that my question, if you do want to offer this to people and say, at least this is something, what claims could you attach to that? What would you say? You probably can't say, here, use this option. We're sure that this is going to get you back. But there's something you could do, right?
Yeah. So I mean, I think that I think first of all, any of these brain preservation techniques, and I include cryonics in this, should be treated for what they are. They are a medical claim. They are you should treat them like a new heart surgery, and that means that the medical community and the medical ethics community must bring to bear their toolbox to decide when and under what circumstances for them to be deployed. The only danger with that is that is if it's applied so naively to say, well, gee, we're going to, since we can't demonstrate that that this absolutely works by having somebody walk off the table, we're not going to look at it at all. And I think that that's essentially what the medical community could say today. They could say, look, this is, we're not even going to think about this because you can't show people walking off the table. But if the medical and neuroscientific communities essentially step back and say, okay, this is this deserves at least enough debate and thought to give a rational explanation for why we shouldn't develop it into a medical procedure. then I'm perfectly okay with that. So, you know, I guess that doesn't answer the question of commercialization, but I don't want to answer any question of commercialization. It's not me. It's not up to me to say what should be commercialized and under what conditions. It's up to the neuroscientific community, the medical community, and the medical ethics community. And right now, all three of those groups have unfortunately dropped the ball on this. They essentially say nothing about this, or they say it's outlandish, it'll never work, trust us. And I think that it's certainly naive to say to dismiss it at that level. I think that it's only dismissed at that level because of philosophical and social issues, and I feel that what those people like myself that really want this to eventually get into hospitals and stuff, what we need to do is we need to just make it very clear that we expect to have a vigorous debate about these issues, and we expect at some point for this to be either rejected on solid scientific grounds, or to be brought in to the medical regulated system in a way that deals directly with the ethical concerns, with the commercial concerns, and all other concerns.
Thank you. Is there any quick follow-on from the room before we move on to the next speaker? Something brief, perhaps? No? Okay.